I'd like just to remind you that we have double entities in the history of life. We have a cultural entity, which is not very old, about 10,000 years. But we have also a biological entity, which is probably coming from a very long evolution. And unfortunately, our cultural system, our brain, is also depending on that biological entity. And I wanted to say that we have lost 3 people we know well: Gennaro Preparata, Jacques Benveniste, and more recently, Emilio Del Giudice, due to the failure of their biological system, their premature failure. And my goal is simply to try to find the factors which make this life so short. And we are now facing new epidemics. We are facing also latent infection and chronic diseases. And this is why I came to water and coherence. So I'm going to speak today on the new findings of this year, but before that I will remind your memory, the brain memory, of what we have done before. We started with a disease which is a chronic disease, but not always, but very often, due to a small bacterium called Borrelia burgdorferi. It is the agent of the Lyme disease, which is transmitted by tick, and sometime, when it's not early treated, give rise to a chronic infection, where it's very difficult to find the germ, but the germ is there. Thank you. And there are 2 technologies we use. One which was very well known, the polymerase chain reaction called PCR. I will show you in a minute the principle. And also a new technology based on the production of electromagnetic waves from the blood of the patient, but also from the DNA extracted from the blood. Classical PCR amplify a fragment of DNA many times because we use a polymerase which is thermoresistant, isolated from a bacterium living in hot springs. It's called the Thermophilus aquaticus polymerase, the TAC polymerase. And using this TAC polymerase many times, because it resists heating, we can open the double helix of the DNA by temperature, by melting. And this melted DNA will be the template for the TAC polymerase, using also some oligonucleotide as primers. And this can be repeated many times because we can melt this first double helix and make another single strand in which the polymerase will work again. So we can amplify several times, thousands times, and millions of molecules by this technology. And we can isolate the fragment amplified called amplicon. The other techniques, which I'll describe in the previous water conference, apply a new property of DNA, of some of the DNA, not all, in water dilution. Water has to be there in dilutions. And the emission could be recorded And the surprise was that many filtrates of bacteria, and also some virus, and also the plasma of patients which were infected with the same agent, but also the plasma of people suffering of chronic diseases, which were not known to be of infectious origin, also show some emission. The initial procedure is very, at least, simple, but also it has to be well obtained, well followed, but once we follow that, it works all the time. We have to work with some dilution of DNA, nanogram amount, and filtration is very important. And filtration is not only to remove, for instance, the microorganisms if we work with culture, but also probably to change the water structure so that it's more open to message coming from the DNA. Also, mechanical shaking is very important, and of course, we ask why we need that. Probably one explanation is the formation of nanobubble. But I heard today from Vladimir that perhaps this also induces some oxidative molecules from the water. So this slide shows you simply the technique in which we dilute in decimal dilutions the DNA solution, filtration at a filter which could retain some nanostructure, and this is very important. What I would like to correlate today this nanostructure of the 100 nanometer side with a coherence domain of 100 nanometers. And we dilute down to 10⁻⁵, and we find in some dilution in yellow, can I have the, in some dilution, the production of signals, electromagnetic signals over the background. And for that, we have to excite the system by external energy coming from a generator or from the natural background, which is mostly the Schumann resonance, which are the surface of the Earth. So we detect signals which have a higher amplitude over the noise, but also a different frequency between 1,000 and 3,000 hertz. So here is the example of the increased amplitude, which we observed first in 2005, and also the analysis of the frequency show an increased frequency on the right over the background here. And this can be analyzed by different Fourier transform here according to the frequency in different color. This is 50 hertz, this is 20,000 hertz, and you see here in the positive signal an increase in this region. The interesting feature is that we don't see the signal in the first low dilution, but only after certain dilution, here 10⁻⁵, and in the following dilution as well. And again, we see the background in the higher dilutions. So those dilutions, I like to stress that they are much lower than so-called homeopathic dilutions. But in the first dilution, probably the DNA is still there, but not in the dilution of 10⁻⁴, 10⁻�13. So probably something else is emitting the signal. The interpretation of this is that the double helix probably change the water structure, perhaps coherent domains, but it's in a lower dilution. There is no emission because of the jam of too many excitation. And only at some dilution, you have the single domains which could emit signals. We show that the electromagnetic signals are indeed produced by the water nanostructure we call nanions. First, from the filtration, their size is over 100 nanometers for the structure coming from bacteria, and smaller than 20 nanometers for the sequence, the electromagnetic signal coming from viral sequences. And work with Mariana Tonsenkova, which is here, indicate that there are some spectrometal change in the near-infrared spectrum in the dilution which are producing electromagnetic signals. But the most important thing is that those structures and also the signals are carrying specific DNA information, including the sequence. In order to show that, we had done several experiments. Those have been mentioned before, so I will be brief, but the idea is to take tube 1, which contains a dilution of DNA emitting signals, put it close to a tube of water, tube 2, in a shell of new metal, so it's not open to the natural electromagnetic noise, but we generate a 7 Hertz frequency, which is the minimal frequency inducing the phenomenon. And the phenomenon will be when the two tubes are put in a coil, there is transmission of the tube 1 to tube 2, both the capacity of emitting EMS, electromagnetic signals, but also the capacity to retrieve the DNA sequence from the tube 1. This is done by PCR. On the top, you have the band corresponding here from the fragment of HIV DNA, which is 104 base pair long. And you find in the tube 2, which have received overnight the information for tube 1, a band which goes even to dilution 8, -8. So this probably also suggests that the PCR by itself is using water memory, and that this would imply that the Taq polymerase will be able to read the structure induced in DNA, in water. In order to respond to some criticism, we design another type of experiments in which we digitize the signals in a computer, and this signal was sent over internet to a receiving laboratory, a distant laboratory, which could retrieve the signals, transform into analog, and put electric current from the signals in a coil surrounding a tube of water, of pure water. When we say pure water, I'm not excluding, of course, there are some impurities which could play also a role. But we use water which is DNA RNA free because we need that for the PCR reaction. And with the PCR ingredient, the Taq polymerase, we could retrieve also the same band and the same DNA. This has been done in several laboratories, including the Benevento laboratory in Italy, and also a laboratory in the University of Göttingen, which I will show you the results. This is done by a young student, so it means It's not very difficult to do the experiment. Within days, he learned the technology. And we can see here, again, in some dilution, the DNA band synthesized from the signals of the water. Of course, one of the main questions that brings me now to the actual work is why we can read DNA induced structure, water structure, with a polymerase, a DNA polymerase. I cannot completely have the answer, but this is really implied in the results. If we believe in the results, we have to imply this. And in order to go further, I ask myself, can we have the same property of the tag polymerase in human cells? Can eukaryotic cells have also polymerase activity which is able to read the water structure? Again, we go back to Borrelia burgdorferi, the 16S ribosomal DNA, and the PCR amplicon, which is 499 base pair. First step was therefore to record that signals and store it in a digital form as a computer file. The second step was to transduce that message from the computer, amplify in a classical amplifier, and use it for passing through a coil surrounding a flask of culture. Here the flask was vertical because we use cell in suspensions, and we expose the signal for several days. It's a loop. The signal, each signal is taken for six seconds, but we expose the flask in a way we will not heat, of course, the flask. They are very low voltage. This is very low energy. And we observe after 4 days some, I would like to say, amazing results. The cell died. Here we show that on the top, the normal cells, the cells which were not exposed to the signals. It was a control. After 4 days, we have in red the number of dead cells, which was very low. But again, we did the same experiments with the cells exposed to the signal. This is time 0. And after 4 days, in the bottom, you find very few living cells, and many cells dead cells. This was a staining, classical staining by blue trypan, which stain only the dead cells. I must say, at the same time, after 4 days, we extract the DNA, and we had the surprise to see the band of the ribosomal Borrelia burgdorferi DNA. After day 4, 5, 6, day 7, the cells were completely dead, and we have this DNA. We centrifuge the culture to see if the band belong to the cells, to the pellet of cells, or to the supernatant, and the band belong to the pellet of cells. So this would mean that living cells are able to read the nanostructure induced in water. This is the control of a noise, a signal coming from the culture medium without cells. We extend this study, of course, to several messages and several types of cells. For instance, after the 16S ribosomal DNA, we use also the 16S ribosomal DNA of a cofactor of HIV, which is present in red blood cells. this DNA can be detected by PCR, but doesn't induce also some cytopathic effect, and we can retrieve the DNA. It's interesting that this cofactor, which is a small bacterium, has picked up some human DNA sequence. And the human DNA sequence also does emit electromagnetic signals and also induce cytopathic effect and also DNA in the cell. So this would mean that the property of bacterial DNA to induce electromagnetic signals is not linked to the sequence. It's linked to the fact that the bacteria is pathogenic and probably induce oxidative stress. There are some reasons to think the property of that DNA is linked to oxidation, and therefore only that DNA which has passed an event of oxidative stress in the patients have this property. The human sequence belonging to the genomic DNA does not emit signal, does not induce cytopathic effect. We also use some DNA, ribosomal DNA of some other bacteria involved in also in pathogenic condition, here autism, Suterella. But this ribosomal DNA does not emit signal, it does not induce cytopathic effect, nor DNA retrieval in the cells. And also the surprise was the long terminal repeat of HIV DNA, which is also emitting signals, does not induce cytopathic effect, nor DNA in cells. So there is some strong correlation between the induction of DNA and the cytopathic effect. The second conclusion is that most of the cells we have been studying are cells of cancer origin. This is the case of two cells growing in suspension, HL16 from a lymphoma, U937 from a leukemia, and both are inducing the cytopathic effect and the DNA. On the contrary, if we use the same signals into normal cells, T lymphocyte or a fibroblastic embryonic cell line, MRC5, normal cell line, we see no cytopathic effect and no DNA. Of course, those results are too preliminary to conclude that we are only inducing this phenomenon in cancer cells. We have to extend, of course, for instance, to stem cells to see if the stem cells behave like the cancer cells or to normal cells. But this result is really impressive in showing that indeed some human cells have the capacity to read nanostructure, coherent domain, and keep the sequence of DNA. So this opened the way to, of course, to many medical applications and also fundamental research, because we are now a very specific DNA we can control. We can control all the steps of this transduction of DNA and see, for instance, if the oxidation of DNA is at the origin of the signals or something else. It seems that there is some kind of tag on the DNA which is, once it's produced, resist all the transformation into EMS, into nanostructure, and indeed the DNA we can retrieve from the cells treated is still emitting EMS. I'd like to finish, if I have still some time, with one medical application of this technology, which I would call digital biology, after Jacques Benveniste. We investigate the role of signals in a disease of children, autism. And to summarize the conclusion of this work, it implies the gut, the blood, and the brain in a microbial connection. Probably there are two important barriers which have been modified in this disease. First, the passage of the gut, some gut microbes to the blood, and also the passage of the microbes of products of this microbe to the brain. I'm not saying this is the only possibility. Of course, there are many factors in autism, but this is a clue and which leads to therapeutics which seems to be efficient. Just to show you an example of a young child who had autism, and we could detect in his blood signals. Here we measure in order to have quantitation, we measure the intensity of the signal, not the frequency, but the frequency was the same as usual. I must say that in our technology, we cannot detect specific signals, specific of the DNA sequence. Probably those signals exist because we can transmit them, but we cannot detect it in our technology. Probably this is also the subject of our further studies. So this child was treated by a large spectrum antibiotics, azithromycin, and we could see after 50 days a decrease of the intensity of the signals, some improvement in the child conditions as shown by his family. And then we could not completely eliminate the signal, so we decided with the clinicians treating the child to change the antibiotic to another one, which induce, again, a further decrease of the signal. This child is not cured, but he was improved by this treatment, and we have many other examples of the same type of curve using the measurement of the EMS, the electromagnetic signal. So this technology, together with the PCR, because in that case, in the case of autism, the main cause of this change in the microflora was the emergence of a new type of species. And this is the work of the group of Jan Lipkin at Columbia University. They designed a PCR to detect this species, and we find some good correlation between the signals production and the PCR. Here are some results with the PCR, indicating most, if not all, children are also positive with this PCR. And AB mentioned antibiotic treatment, and you can see here, the same child I showed you before, that after 5 months, the band of DNA has decreased, but not completely disappeared. And the same for two other children in which the antibiotic treatment decreased, but not completely eliminate the bacterial DNA. So to summarize this study, over 150 children in a cohort, which was done mostly in Italy, France, and Belgium, we see that 85% of those children have electromagnetic signals in the DNA of their plasma, and 82% are positive for the Suturella PCR, meaning that some children have probably are also infected with other bacteria which are not recognized by the PCR, which is highly specific. We also did a study with another bacterium, PCR Borrelia, and the results were not, the difference were not significant compared to the group of healthy children. But here you have only 3% positive for the signal and 9% for the PCR citérella. So this indicates that this technology has some practical application, which is to follow up monitoring some therapeutic treatments. And this model of transmission from the gut to the brain probably could be explained also in other neurological diseases. It's not only restricted to autism. So this is a model I like to propose in which there is a continuous expansion of some bacterial agents due to some genetic factors, environment, and this induces partly immune suppression, oxidative stress, which in turn, of course, induce more bacterial agent latency, infections, oxidative stress, and finally, oxidative stress, free radicals will attack the DNA, induce mutation, and those mutations may fix the disease, the symptom of the disease, without excluding the possibility that at the end there are some transmission from modified proteins to proteins, which is the prion effect, and of course those phases will be irreversible. So this model suggests that we have to act early, as early as possible, and probably the detection of electromagnetic signals may be one of the first signs of the infection, of the latent infection, and this is where we can act to reverse the disease to a normal status. This is our hope, and of course, I'm very impressed by what I heard today on the expedition. I am also, of course, on the ground, on the biological aspect of us, but I was very impressed by the fact that I have now a coherent vision, thanks to our colleagues, physicists, and the first one was Emilio, a very good idea of what's going on in water. Water is probably the medium which is has a memory, a flexible memory, so it could go back and forth, and like the basic memory, which is now DNA, but one cannot exclude that the origin of life, water was first, and DNA or RNA came later. Thank you.